Thank you guys for joining. My name is Charlie Nunn. I'm at Duke University in evolutionary anthropology and the Duke Global Health Institute, and I run the Triangle Center for Evolutionary Medicine. And this is Club Ev Med. Club Ev Med is a semi-regular meeting a couple times a month where we highlight new research in evolutionary medicine and just try to bring the community together and learn about advances in evolutionary medicine. TRISEM's organized actually by a bunch of different evolutionary medicine centers, and I've just pasted that into the chat. And that includes the Triangle Center for Evolutionary Medicine here at Duke, UNC, NC State, NCANT, NC Central. Good to see some of the people from those institutions attending today. We also organize with the International Society for Evolution Medicine and Public Health. And I just want to encourage everybody to please consider coming to the annual conference. It's this year, July 8th to 10th in Nashville. The program is really shaping up to be a great program, and we hope to see a lot of you there. And in addition, I want to call your attention to the Society's journal, Evolution Medicine and Public Health. Again, you have a link in the chat that can get you there. Okay, well, I think we'll just go ahead and get started with the rest of the program. And it's really a pleasure today to introduce our two speakers. We have Nathan Crook, who's an assistant professor at NC State. He's in the Department of Chemical and Biomedical Engineering there. His research focuses on the human gut microbiome, and he's really operating at this kind of intersection of research on metabolic engineering, synthetic biology, and microbial ecology. And I like that ecological side. And he also is very interested and competent in the evolutionary side as well. One of his lab's goals is to design and assemble microbial communities to enable the conversion of food into a healthy mix of energy and nutrients and therapeutics, and really thinking about the ways that they can harness the power of the microbiome. He got his PhD in chemical engineering at the University of Texas, and before that, a Bachelor of Science in Chemical Engineering at the California Institute of Technology. And he's received many awards, including the NSF Early Career Development Award from the NSF in 2023, and then the next year an NIH New Innovator Award in 2024. Nathan gave a TRISEM talk. We hold these meetings in the Triangle of North Carolina, breakfast talk, and he gave a great breakfast talk. I wanted to include a microbiome talk in Club Ev Med before the year was over, and he immediately came to mind because he presented such an innovative set of research, very rigorous and innovative approach to his research. And with him today, we have Alexandra Imre. She's a postdoctoral researcher in the Crook Lab at NC State. She's been here for about 2 years. She has a very impressive academic record with a Bachelor of Science, a Master's, and a PhD from the University of Debrecen in Hungary. She's mostly worked on fungal microbes, has a very nicely growing, very impressive CV, a collection of publications focused mostly on fungi. And as I said, she's been here for about 2 years. So I'll turn things over to you guys. Thanks so much for joining us. Great to have you here.
Thank you. Thank you for inviting me. This is an honor to present to all of you and talk about the research that we're doing. What I decided to do today, as I sort of get my screen shared, was to have kind of a two-part presentation talking about the work that I did as a sort of a postdoc that ignited my passion for this idea of in vivo evolution of live microbial therapeutics. And as a postdoc, I did this in E. coli, and Alexander will discuss where we're taking that right now in our lab. And so the title of my talk is about probiotic E. coli, and I wanted to give a shout out to the people who worked with me during this talk back at WashU. This is us setting up our own germ-free mouse facility at WashU. We have our isolators here, and my PI at the bottom is Captain Dantas. So it was a really great experience setting all of this up. We were guided by this sort of conundrum, right? If you have a probiotic that you're going to deliver to a person, it experiences 2 distinct life stages, one of which is that it grows in a bioreactor or a food culture. And in that environment, it's very nutrient rich, right? It's almost the only game in town. There's not many other microbes that are present. So there's, the nutrients are around and perhaps not as much selective pressure for things to happen. On the contrary, that gets then delivered to your large intestine, where there's a variety of diverse selective pressures operating on this microbe. There's the flow of material trying to push this microbe out of the intestine. There's the host immune system. There's maybe bacteriophages. There might be metabolic interactions between existing microbes that this probiotic then has to go into. Now, because of these alternating selective pressures, we thought maybe there's a drive for evolution of these live microbial therapeutics in the intestine. So our questions were, number one, do probiotics evolve when they're in the gut? Number two, if they do evolve, which functions are under selective pressure? And then number three, how does this depend on their environment? The diet, the microbiome state, et cetera. How do those impact which things are recovered? So we had this question and we wanted to study it in the context of this probiotic called E. coli Nissl 1917. This was discovered by Alfred Nissl in 1917, of course, as the name suggests. And it's sold in Europe as Mutaflor. It's had efficacy against IBD, diarrhea, and constipation. For us as engineers, though, it was very interesting because there are a growing number of startups interested in using this microbe because it's an E. coli strain. It's easy to engineer. You could use it as a chassis for engineered function. They try to put new genes in and get it to do a thing in the gut. Actually, famously, recently, Synlogic had attempted to use this as one of their chassis for their therapeutics. So within the context of this microbe, we had a few different things we were trying to vary. The first of which was the microbiome state. We did experiments in germ-free mice, so it was monocolonized with E. coli Nissl. We had a defined community of microbes, so a handful of microbes that were there, even all the way up to conventional mice that had a very diverse microbiome. We also tested the different diets of those mice. And then finally, different body sites in the intestine. We looked for where these tended to live, because those could also exert different selective pressures. We also looked in the context of this habitat variation, different types of evolution that might be happening. Number one was what we had these different scales of evolution in microbiomes that we talked about in a review article. One was within genome evolution, so a microbe using the DNA within its own genome as substrate for evolution. We have their SNPs or insertion of native sort of insertion sequences, for example. Alternatively, you can imagine microbes sharing DNA within the microbiome. Maybe there's conjugation that occurs and shares DNA within these microbes. And so we had these two different ways in which microbes could evolve, and we used two different ways to analyze this. So I'll talk about scale number 2 first. How would we identify genes that got transferred between microbes in the human gut? So we did this in the following way. We used functional metagenomics to do this. Functional metagenomics kind of forces horizontal gene transfer. So we weren't looking at native rates, but we were looking to see if horizontal gene transfer happened, what genes would be selected for. The way we did this is we took fecal samples from bacteria that were already in the gut microbiome. These were fecal samples from infants and also infants' mothers. So either a developing microbiome or a already developed microbiome. We extracted the DNA from those fecal samples and we chopped them up into small kind of gene-sized or small operon-sized bits, 2 to 5 kilobases in size. What we then did is ligated that DNA to E. coli Nissl, right? So we put those in expression vectors and made a big library, upwards of 10 to the 6 different clones of E. coli Nissl, all expressing a different fragment of metagenomic DNA. Once we had this library, we could then deliver that to the mice of interest. And those that were able to colonize would rise in abundance. Those that were not able to colonize very well would decrease in abundance. So out of that 10 to the 6, then the ones that could colonize well would be enriched. And then we could sequence those, annotate them, and figure out what the functions were. And hopefully if this all worked out as we expected, we get going from a 10 to the 7 gene pool down to maybe 1 to 10 genes that were sort of putative colonization factors. And again, the notion here is not to measure the rates of horizontal gene transfer, but rather what functions would be selected for if they could undergo transfer. What we first did is we looked at germ-free mice and we did different types of diets. Here's a graph of different abundances of inserts as a function of time. On the y-axis is the abundance of that insert, and it's a stacked plot, so the overall number should add up to 100%. And on the x-axis is the day of the experiment. So on day 0, we delivered 10 to the 7 gene fragments. They're much too small to plot, so we only plot the ones that had a high abundance in this study. But over the course of 15 days, you get this coalescence of diversity down from 10 to the 7 down to maybe a handful up to 5 fragments in this particular mouse. This mouse was consuming standard mouse chow, right? So the plant polysaccharide based diet. We also had mice that were consuming what we call the human diet. This is really more like a Western diet, high fat, high carb contents. And that human diet was placed in the context of either it by itself or several different prebiotic fiber that we added to that diet as well. Here's what I'm showing here are the abundances for one particular mouse, but then we have below this graph replicates from the other mice in the same cage. And what you see is that the microbiome and the insert abundance profiles are relatively the same between these different mice that we had in those studies. So that was reproducible within that experiment. We took the genes that we got, the sort of the most highly abundant gene fragments, and we sequenced them. We were most interested in these mouse diet fragments because they were highly reproducible, but also there was this one fragment, human diet 4, that was the same across all of the human diet arms that we had, no matter what the inulin, no matter what prebiotic we had. We sequenced them and we got the following sort of architecture of that insert, and we colored the gene based on what the predicted function was. In blue are the glycosyl hydrolase family 32, or sorry, purple. Those we saw in many different of our inserts. So we thought those were kind of important. We wanted to go into them a bit further. Glycosyl hydrolase family 32, if you know anything about glycosyl hydrolases, that's involved in consuming sucrose. So this was a gene that allowed E. coli to consume sucrose, we think. We took these gene fragments and we cultured them in the presence of a variety of polysaccharides that contained sucrose. Number one was sucrose by itself. Another one was raffinose, stachyose. One of them actually enabled our cell to grow on galactose. And then another one, we grew them in sort of a liquid homogenized version of the diet that we were giving those mice. And it turned out that these were actually able to grow quite well in that environment, indicating that the presence of sucrose in the diet was a carbon source that these E. coli wanted to be able to. We did a similar sort of experiment in non-germ-free mice. These mice contained a defined community inside them. These were the microbes that we chose that were sort of representative of the gut environment. What we then saw show up in those types of mice was this following a few examples of this insert that we had. These were actually fragments that we got from E. coli. They were, the whole microbiome was there to choose from, but this particular gene fragment was sourced and it was homologous to the E. coli's own genome. Now, this was overexpression of a transcription factor in pink. And this particular transcription factor was GNTR. So it's a repressor of gluconate consumption. If you overexpress a repressor, then it doesn't eat gluconate as well as it used to. And here what you're seeing is that E. coli that is expressing one of these 4 inserts does not grow very well in gluconate, but grows just fine in glucose minimal media, indicating that for some reason it was advantageous for these E. coli to not eat gluconate. This confuses us to this day. Other evidence from other researchers indicates that gluconate is actually an important carbon source for E. coli when it's in the gut. Potentially in this community, there's something special about it that makes gluconate consumption disadvantageous. So moving on to scale what we saw within genome evolution, we needed to look at whole E. coli genomes. And so just to see what we did, we took the fecal samples from those experiments and we plated them on Petri dishes and picked single colonies. What we then did is did a combination of long read and short read sequencing. This allowed us to get, number one, a closed E. coli nissl genome that we could then use as a template to look for mutations, but then also 400 whole genome sequences that we could then check for conserved mutations that often occurred in our experiments. I'll show you a couple of them here. And what you're seeing here is sort of a wheel showing all of our 400 isolates. And then the different concentric rings have different sort of metadata corresponding here. So the innermost ring is the mouse that this isolate was derived from. The second ring is the particular microbiome background, whether it's germ-free, a defined community, or conventional mouse. The diet is the next ring, tells you where it came from. And then it's particular type of mutation we identified in those isolates. So there's a lot of data here. The important takeaway here is that we saw multiple cases of GNTT being mutated. These tended to be premature stop codons. These amino acid going to a star indicates that it was the insertion of a stop codon in that gene. This was also a missense mutation that we think is detrimental. What's going on here is that GNTT is a transporter for gluconate. And in this case as well, these strains, in addition to having an insert that prevented the transcription of their gluconate utilization operon, they also had mutations in a transporter for gluconate. So we again, additional evidence here that gluconate was bad for the E. coli. Another thing that we saw was increased growth on a different sugar, N-acetylglucosamine. N-acetylglucosamine is a major component of mucins, and we saw mutations in this gene called NAGC. These were also largely loss of function mutations in NAGC. A lot of stop codons, premature stop codons, frame shifts, as well as a changing of the start codon to a non-start codon. In this case, what we saw from that gene was that mutants containing these mutations grew better on N-acetylglucosamine. They grew to a higher final cell density than the starting points did. One of the last things we saw that I'll discuss here is when we did what we call a streptomycin-treated mouse model to kind of repress the native microbiome to allow E. coli to colonize at a higher level. When we gave E. coli Nissl to those mice, even though it was after the time of streptomycin dosage, we saw a preponderance of mutations in this gene called RSMG, which has been previously known to confer streptomycin resistance to E. coli. So even though these mice were not taking streptomycin at that time, they still had presumably antibiotics in their gut that allowed a selective pressure for these mutations to occur. And so just evidence that these mutations do cause streptomycin resistance is in this paper down below. So we think that this is an interesting work because it indicates that live biotherapeutics can evolve right when they're in your gut. They do things that are, to our knowledge, not explicitly pathogenic in nature. So they're not trying to cause E. coli to become a pathogenic E. coli, but rather to allow it to, number one, consume carbohydrates. Number two, we saw mutations for increased epithelial cell adherence, and then also the resistance to antibiotics that were administered. So they're just trying to survive in this new environment they're placed into. Hopefully, as we explore this question further, it motivates others to look at evolution of live biotherapeutics in sort of in a clinical setting. So that's my half of the talk. I want to thank the folks that did the work at WashU. I have a picture of my current lab down below who is sort of carrying on the torch of microbiome engineering. And with that, I'll stop my presentation, give it to Alexandra to talk about what she's doing.
All right. Thank you, Nathan. It was a great talk. Okay. So first of all, I want to greet everybody. So thank you for having me, and thank you, Charles, for the amazing introduction. First, I'm just okay. So during my presentation, I would like to talk mostly about the adaptation strategies of Saccharomyces boulardii probiotic yeast in mouse gastrointestinal tract. So compared to Nathan's talk, now I would like to shift from bacterial probiotics towards the yeast probiotic. And what is important to know about the Saccharomyces boulardii probiotic yeast? So the Saccharomyces boulardii probiotic yeast is on the market since the 1950s, actually. And it is becoming recently a very popular probiotic due to a number of reasons. So one of the reasons is its health effects. So there are numerous health effects, positive health effects of Saccharomyces boulardii probiotic yeast, including antibacterial effects, immune modulation, and other trophic effects in the GI tract. And just to mention a few, it produces enzymes that can cleave Clostridioides difficile toxins. It adheres to pathogens, produces acetic acid, induces anti-inflammatory cytokines, and etc. It's also becoming very popular because the administration of this probiotic is very convenient. It can be taken at the same time with antibiotics. And in hospitals, it is administered to prevent Clostridioides difficile infections. And what is very important to mention now is that the genetic engineering of this probiotic is also becoming popular, mainly to develop new strains to increase beneficial health effects and also to increase colonization. And this is going to be the main focus of my talk, because it's a good thing to increase colonization, because we can increase the effect of the added health effects that we genetically engineered into the probiotic yeast. So we can prolong those positive effects. However, our information of the adaptation of this microbe in the gut is limited. So it can also happen that we take this probiotic, it resides in the gut for a longer time, let's say, and it adapts in a way that can even interferes with our initial so it can interfere with the health effect that we engineered into the yeast. So we try to identify the adaptation mechanisms of this yeast because of these reasons. So we designed an experiment where we gavaged germ-free mice with this yeast, and we collected fecal samples at week 1, week 3, week 5, and week 6. It's important to mention that the residency time, the normal residency time of Saccharomyces boulardii, when normal microbiome is present, is 3 to 5 days. So this experimental design, by using germ-free mice, allowed us to gavage the mice only one time at the beginning of the experiment, and the yeast could reside in the gut for 6 weeks. So from the fecal samples, we plated this yeast, re-isolated them, and applied phenotyping experiments and also genomics. So for now, in this talk, I will focus on the spot plate stress phenotyping and the invasivity assay that we did, and also the genomics. I applied the GATK genomics pipeline, and at the end of the genomics pipeline, I used the SNPF program to identify high-effect mutations in protein-coding genes. So in case of the spot-based stress phenotyping experiment, I focused on to characterize the yeast in case of the presence of high temperature, and also to test their tolerance to different stressors. In case of the high temperature, it was interesting to see that both on YPD and synthetically defined media, the yeast showed normal growth at 30 and 37 degrees, but many of the isolates did not show growth at 42 degrees of Celsius. The tolerance towards stressors was variable, but what is more important to say is that compared to the Y-type strain, we observed lower amphotericin B and Congo rat tolerance, and that indicates SAVO remodulation in this gastrointestinal in these isolated yeast, and also we observe lower tolerance to high temperature. So interestingly, these results can suggest adaptation to environmental conditions that did not pose significant stress to the probiotic yeast, which kind of makes sense, because if you think about that, in our current experimental setup, there was no microbiome that could pose stress to the yeast cells. And other than higher temperature, which is 37 degrees in the mice, there is a high chance that there was no stressful conditions that these yeast needed to adapt to. During the invasivity assay, I used 4 different agar plates. It's important also to mention that invasivity is, in case in the literature, at least based on the literature, invasivity in case of the yeast occurs in case of the lack of nutrients in the environment. So that's why I use YPD and synthetic defined media, which both provides the essential and all the nutrients that the yeast needs to grow. But I also applied media that has a low ammonium content or low glucose content. So overall, what I could see is that there was no invasivity at 37 degrees. However, compared to the Y-type, there was increased invasivity in case of the mouse GI tract isolates at 30 degrees, which you can see here on this figure. The phylogenetic network that I built based on the genomics analysis was showing that towards the end of the experiment, the week 6 isolates has a higher genetic distance compared to the samples that were taken at the earlier time points of the experiment. So on this figure, you can see the different colors that corresponds to the time points that we took these samples. The green dots representing the isolates from of the week 6 isolates, and as you can see on this phylogenetic network, they are more far away from the other isolates. So this was also showing a clear. This shows us a clear sign of adaptation. I also I also investigated the chromosome coverage after sequencing this yeast. And this chromosome coverage actually showed increased frequency of the chromosome 16 trisomy, which should suggest adaptation to heat stress, which is also very well described in literature. So just to explain it a little bit more, so how the mechanisms how yeast adapts to, for example, high temperature, but it also can be true to other environmental factors. First, there is what's happening is a so-called aneuploidy-assisted adaptation. So first, not actually mutations happen, but first aneuploidy appears, and this is what we see. So aneuploidy is the difference in the numbers of the different chromosomes. And then after a couple of dozen generations, the ploidy restores, and then compensatory mutations also appears that actually fixes the adaptation towards that in that environment that the yeast experiences. These figures are so-called allele plots. So these allele plots are important to look at because it helps us to identify, other than the chromosome coverage, helps us to identify other bigger chromosomal rearrangements in a genome. So as you can see on the chromosome coverage map, you can see that the trisomy of the chromosome 15, and that is also visible on the allele plot because of the division of the dots on the figure. However, loss of heterozygosity cannot be observed on the chromosome coverage figures, but we can see it on the allele plots. So these dots actually represent the heterogeneous positions in the genome. And when we see, for example, the lack of them compared to the wild type, that is an indication of loss of heterozygosity. Other chromosomal rearrangements that can be observed on the allele plots are segmental duplications, which we also have seen in a number of chromosomes. In this case, right at the beginning of the chromosome 4, there was a segmental duplication. So based on these allele plots, actually we could identify 5 different chromosomal rearrangements and their frequency was different in case of the different at the different time points of the experiment. So it was very nice to see that towards the end of the experiment, the frequency of these bigger chromosomal rearrangements actually increased. So as you can see in the case of the week 6 isolates, there was actually 18 isolates that were affected by the chromosome 15 trisomy, and this was the most important change that we saw. But also in case of other chromosomal rearrangements, it's clearly visible that these aneuploidies and gross chromosomal rearrangements increase towards the end of the experiment. We also identified the genes that were affected by high effect mutations. So in our case, high effect mutations mean the mutations that results in the loss of function of a certain gene. In our experiments, we identified 19 of such mutations, so 19 genes that were affected of such mutations. And the majority of such mutations appeared in the week 6 isolates. And just some, to just mention some important ones that we observed. So for example, the RGD1 gene is involved in repolarization of actin cytoskeleton during adaptation to heat shock stress. So the repolarization of actin cytoskeleton is actually required for the cells to be invasive. So if you remember on my previous slide, that we observed an increased invasivity that can be due to the effect of this mutation, for example. Another mutation occurred in the CYC2 gene, 14 isolates of the week 6 isolates were affected. So it was almost half of the yeast that were affected by these mutations. The loss of function of this gene results in the decrease in competitive fitness. This was also this was also observed in our phenotypic experiments, but decreased resistance to chemicals, also decreased stress resistance and increased heat tolerance. I have to go further and investigate this one, the increased heat tolerance, but overall, these results aligns well with the phenotypic experiments. The IRA2 gene required for reducing CAMP levels under nutrient-limiting conditions. So again, the GI tract poses a nutrient-limiting environment when the yeast is present there. So this mutation also indicates adaptation towards nutrient-limiting conditions, and the WHI2 gene mutation is actually also has a role in nutrient depletion environment, and it's also required for the activation of general stress response. So just to summarize our results, so spot-based stress phenotyping revealed decreased tolerance to chemicals and heat stress, indicating that conditions in the GI tract were not posing significant stress to the probiotic yeast. So the yeast didn't need to maintain the genes that are required for stress tolerance. And invasivity actually revealed increased invasivity of the GI tract isolates at 30 degrees. It might be treated due to adaptation, but due to adaptation, because it's very interesting that we haven't seen this adaptation at 37 degrees, which is actually the temperature that occurs in mice also, not only people. And the genomics analysis showed increased chromosome 16 trisomy. It could be due to heat stress adaptation, but it can be a result of other environmental factors too. And the appearance of chromosomal rearrangements are plastic changes due to adaptation. High effect mutations in genes that has a role in chemical resistance, actin repolarization, and activation of general stress response were also observed, and that might explain the high level of invasivity and decreased tolerance to chemicals in our isolates. And it's still an ongoing research, so it's maybe very early to say any general conclusion based on these experiments, because we also are planning to test for colonization, both in vivo, both using an in vivo and in vitro model, to test if these yeast isolates are indeed reside in the gut more time than the wild type strain that we used at the beginning of the experiment. And thank you all for your attention. And I would like to thank Dr. Nathan Crook I would like to thank Nathan for supporting me during this project, and also our collaborator, and who was at the same time my previous supervisor earlier during my PhD, Dr. Walter Fligler. And I would like to thank also the undergraduate students who are assisting me during this project, Kelly Abernathy and Kyle Kay.
Okay, thank you, Alexandra. Thank you, Nathan. Fascinating, really interesting work. Thank you for a great presentation. We have at least one question in the chat. Paul, would you like to proceed with your question?
Sure, thanks. That was really interesting work. I have a couple of questions and a comment. I guess my first question is, it seems like from the tables that there was a dramatic change in the number of chromosomal rearrangements from week 5 to week 6. Do you have some hypothesis as to why you might have observed this? Is there something critical about that time period?
Yeah. Alexandra, you're muted.
Yeah, thank you. Sorry. So I don't think that it can be. So it's. Oh, you mean there was a significant decrease compared to week 5 and week 6? That's what you were asking
Yeah,
about.
it seems like there were more, like your table of chromosomal rearrangements. The numbers went up quite a bit. I mean,
quite a bit. Yeah.
I think from like 8 to 18 in terms of the trisomy. It seems like there were some other ones there where with very low numbers and then.
Yes. So I think what could have happened, and actually I kind of mentioned this. So first, how yeast adapts is it applies a series of plastic adaptations, And we could see these segmented applications and loss of heterozygosity, and we actually could see a number of segmented applications, but those changes are very plastic. They appear, disappear. And what happens when yeast experiences a certain environment is that when the longer the time, it tries to adapt. So these are short-term adaptations, but there are more fixed, more long-term adaptations. And what could have happened is by week 6, the yeast cell needed, I have to phrase it like this, like needed to adapt in a way that's more fixed. And it seems like one way to adapt to this environment that was that we applied to the yeast is to increase the chromosome 16 and chromosome 15 trisomy, and also on that chromosome, what I could see is an increase of single nucleotide polymorphisms. So it could have happened that actually it took 6 weeks for the yeast to kind of decide what mutations are going to be beneficial in that environment.
And my comment was, I noticed IRA1 and IRA2 on your list. Those are among the most commonly hit genes in Saccharomyces cerevisiae experimental evolution studies, which are again often typically done in nutrient limitation. And in fact, we've got sort of a pet hypothesis that cyclic AMP signaling is sort of an evolutionary hotspot for yeast and fungi in general. So that's pretty interesting to see. And then I guess that got me thinking. So I noticed you put Boulardii in quotes. What's the history of Boulardii? Is this an engineered strain created as some sort of hybrid cross between Cerevisiae and some other Saccharomyces? What is this? Is this a real species, or is this an engineered strain that people use?
Yeah. So this is actually a very complex question, but what I can say is there is a story behind Saccharomyces boulardii. Just very short, there was a French scientist who traveled to Indochina, and he isolated this yeast in the early 1900s, and then he deposited it, that at least this is a story, he deposited it, and then in the 1950s, there was a French company who took this strain and put it on the market. In case of from the genome's perspective, it is Saccharomyces cerevisiae. It's actually can be considered a subtype of Saccharomyces cerevisiae. So just very similarly to the wine yeast or baker's yeast, so they are not and in fact, based on earlier researches when people used applied a great genome sequencing projects, Saccharomyces boulardii grouped was the part of the group of wine yeast. So they have said their own subgroup within the wine yeast, but they are very close to the wine yeast. So it's for sure not a different species compared to Saccharomyces cerevisiae. And also it can be still a debate if, the name. So actually the name is not valid. That's why I like to put those marks in case of the boulardii word, because it's not that subspecies, the species was not described according to the rules how we should according to the taxonomic rules how we should name yeast strains. But it's no reason to say that it is a different strain or it is a separate strain from Cerevisiae.
Okay, so it sounds like it's one of those outcrossed wine isolates that are
yes
common. Okay, thanks.
Nathan and Alexander, I had a quick question. I assume if people are taking these probiotics, they want to have them, the ideal is to have them colonize the gut, right? And what you're bringing up is that there seem to be different conditions under which they're produced and what's selected into the production process than what the gut looks like. For example, Nathan, I think you brought up that there's evidence for selection on increased epithelial adherence, right? I imagine that's the environment in which they're produced is not favoring that. Correct me if I'm wrong on all this, but is part of the goal here then to try to figure out, what ways to produce these microbes or, a different way to produce these microbes so that it increases the probability of colonization? Or Yeah, I don't know. I don't know if that's really a question or a comment, but
I think it's a
how
great
are you thinking
idea.
about that?
I mean, it's a clever solution, right? So make the production environment the same as their use case. Right. And so that way
Or at least
there's not
select
this
for the
other
same
pressure.
things in some ways, even if the environment's not the same.
Right. I will say that there are different schools of thought on whether you want probiotics to colonize or not. Some
Uh,
folks are like, get the job done, get out, or like take it once a day, and that way once it's done, and you don't have it hanging around longer than you need to. Others are of the opinion that, if you have a chronic disease, maybe you want something that's there for a long time, intimately associated with your gut, so that it's producing the thing that you want it to produce at that site. So
yeah.
I think both have options, but yeah.
Alexander, do you have any thoughts about that in terms of the production, conditions versus the gut conditions? Oh, I think you're muted. Sorry, I think you were muted, Alexander.
Oh yeah. So can you ask the question again? Because maybe I missed that.
Oh, I was just, I was just curious about the conditions under which these organisms are produced,
Mm,
which are obviously very different from those in the gut. And if the goal for a particular probiotic is to have it colonize the host, does this give any insights into the production side of it so that they, at least the environment in which, they're grown, cultured, whatever, is, more similar or favors characteristics you want, that
good
are favored in the
question.
gut, basically.
Yeah, in case of yeast, I'm not sure about the production environment because these companies, this is confidential information, so they don't disclose these. But it would, for example, in case of the yeast, what I would be interested in if the production occurs at 30 degrees or at 37 degrees. So do they kind of pre-adapt the yeast to the human temperature? That could mean a huge difference because even if in my experiments show that if I apply different temperatures, actually the gene expression and the yeast phenotype changes. So it can also change the probiotic effect even. So the pre-culturing and the conditions for sure could be very important, but I don't think we have a lot of oversight on this because we don't have that knowledge.
I was going to follow up on Charlie's question. This is something I've been talking about probably for at least a decade. It's what I call the off-the-shelf fallacy, in which people just pull something out and they don't actually think about the conditions under which that organism evolved. And therefore, they just throw it into wherever it's going to go, assuming it's going to be able to adapt to these new conditions, it's fallacious. In fact, Nathan talked about the fact that a lot of people think that so-called probiotics don't actually do anything. And most of the literature that I've seen indicates that they do absolutely nothing. And I would argue that that's precisely because they've never really addressed this question of whether they're going to do what you do in production in the environment they're being put into. And of course, you also have to think it from the point of view of the companies. They don't really care if this stuff works. They only care if people think that these things work. And so therefore, their issue is mass production. And the conditions that will allow mass production of these microbes are definitely not the ones they're going to see in an actual human body. So I agree that this is problematic, and you guys are doing great work. I really enjoyed these talks. The only thing I didn't enjoy was the traditional colonizer language, because quite frankly, it has a specific, socio-political history, as opposed to using terms, like establishing itself in the gut or so forth. So I think, people need to give thought to those things. Okay. But otherwise, I think it's great work.
Thanks, Joe. I think I remember in a recent grant proposal, I ended up using like load, right? Instead of colonization level, it was load. And that seemed to go a bit better with my ecology colleagues than that. So I think it's a point well taken.
Yeah.
Okay, so far we have a very TRISEM focused discussion here with Paul from Duke, Paul McGuenny, and then Joe from A&T, and of course our speakers from NCA&T. I was for NC State. Sorry, Nathan, Alexandra. Any other questions? We'd love to hear from some other centers who are represented here and other people. And if not, I'm going to ask another question. So please speak up.
I guess I have one.
Excellent. Go ahead.
I'm Owen. This is actually my first club EdMed. I'm outside the Triangle. I'm in Nashville, Tennessee, at Vanderbilt.
Great.
I was wondering, kind of along those lines of like, how can we maybe make the production environment reflect the selective environment that these organisms are going to see inside their host or the patient or individual taking them? Have you guys put any thought into how like the non-adaptive forces of evolution are going to be impacted by similar things? So, does the anaerobic environment that they're going to see in the gut, does that, or the nutrients they're using shift the mutation rate and spectrum, and things like that? Just dumping them, obviously to a more complex ecosystem clearly is going to allow for more recombination to occur, right, between more distant lineages. How do before we even get to adaptation, have you thought about how much weird diversity that we might not really understand from in vitro experiments might arise?
Yeah, it's a great question. The answer is no. we haven't considered the innate enhancement of mutation rates in stressful conditions as part of this. We tend to look at things that happen to be selected for, right? So it was the stuff that happened and then ended up being fixed. I thought I knew where you were going in that question with non-adaptive evolution and I thought of transcriptomic changes. And we have been looking at transcriptome changes of these microbes when they're in the large intestine, which is of course non-evolutionary process, but it's a totally different mechanism of adaptation there. So no, unfortunately.
Thanks.
Okay, thank you. Other questions? While people are thinking about that, I'll ask my other question. And you might have said this, and I'm sorry, Nathan or Alexandra. Does a more diverse initial microbiome in these experiments with these mice, like Nathan, you had these different, initial conditions. Does that influence the establishment success? And does it show greater evidence for evolution when there's a more diverse initial microbiome? I'm sorry if I missed that.
Yeah,
but
no, Alexandra, did you want to discuss that?
No,
Yes,
I just you can go ahead. You can go ahead.
Oh, okay. Well, I'll tell you what it was for E. coli. In those cases, we saw that the more diverse the microbiome was, the more different microbes there were, the less likely it was E. coli would take a foothold in the microbiome. And so there was this trade-off between diversity and then establishment success there. And so when we did the more diverse microbiome, we saw actually less, I would say, consolidation of functions, if that makes sense, in our evolution. So we saw a greater diversity of things that came out of that mouse we saw less sort of, with the germ-free mouse, you saw like multiple isolates at the same mutation in different mice, right? We saw, or multiple different mutations of the same gene, right? And so there's a very, I would say, uniform selective pressure. What we thought was going on with the more diverse microbiome was that there's different niches that these E. coli's could potentially adapt toward, and there wasn't a consistent, I would say, winner in that under those experiments. So I think that was going on for the E. coli. Alexandra, I don't know if you have any thoughts on Boulardii.
So I think, what I can just add to this is maybe the presence of the microbiome has an effect on the, like as Nathan said, the grip, like how it can adapt to the GI tract and how long it can stay in the GI tract. So in the presence of microbiome, Saccharomyces boulardii actually clears out from the GI tract within 3 to 5 days. So, and the lack of microbiome actually ensures the yeast to stay and more time in the GI tract That's I think the only thing I know. I'm not aware of any research that would have focused on the thing you just asked, like in a more in-depth manner.
Okay, Paul McGuiney had another question. Are there any interesting allele-specific expression differences in S. boulardii?
So in our experiments, like the one I just presented, we didn't look at that, but maybe Nathan, there was another project that Ganan was working on with transcriptomics. So maybe you can have some more
Yeah,
information on that.
I was trying to think. We did bulk RNA-seq on stool samples that contain boulardii in them, and we have not yet looked at whether different alleles are present at different levels. At least not yet. It's mostly just been the individual gene level. But I do know that there are expression differences between Cerevisiae and boulardii that can happen. One time we put a promoter that was the ALD6 promoter from Cerevisiae into boulardii, and ALD6 is normally a weakish promoter in Cerevisiae, but it turned out to be a really strong promoter in boulardii. And so there were these sort of strain specific differences, but we haven't gone down to the LDO level yet. But it's a really cool question.
Just one follow-up question. Do you mostly recover in the yeast experiment? Do you mostly recover yeast cells or do you get spores? Have you tried to look at that in terms of like what's coming out in the waste?
Also, good
Correlating?
question. We have no way of distinguishing those, I don't think, because we plate them directly on nutrient media, and we say, okay, does it have, we put antibiotics on that media, and then we also sometimes will integrate an antifungal resistance gene into the genome of the yeast to prevent other fungi from coming up on our plates. So if they were spores or if they were live cells, we don't know.
Very cool.
Okay, are there any other questions? We have time for one last quick one if somebody has one. Okay, let's thank Nathan and Alexandra for a great talk, and thank you all for the useful questions, valuable questions, and valuable discussion. I want to announce that we have, I think, the last Club of Med coming up next Wednesday. Jennifer, are there any other after that, or is that our last one?
Nope, that's our last one next week. I'll send out the announcement right after this.
Okay. It's going to be a great talk. It's going to focus on Peto's paradox, paper that just came out in PNAS by Chris Venditti and others. It's going to feature Chris Venditti and George Butler are going to talk about it. The title of the paper is No Evidence for Peto's Paradox. This relates to the association between body size and cancer rates across animals. And so it should be a pretty interesting and hopefully provocative talk. Hope you can make it next Wednesday at noon Eastern time. Okay, thanks again, Nathan and Alexandra, and thanks everybody for tuning in.
Thank you very much. This is really fun.
Thank you. Bye.
Bye.